everyone, we are back again talking about the plasma membrane and its importance to the cell and its functioning of everyday life and as its own unique living organism if we're talking about single-celled organisms. Today we're going to focus on chapters 5.3 to 5.6, which is all about passive transport. So just some vocab to help us kind of learn what passive transport is and its importance. So first is diffusion. Diffusion is a type of passive transport. It's the random movement of particles that results in the net movement of a substance from areas where there is a lot of that particular substance or of high concentration to areas of low concentration where there's not a lot of that particular substance. A concentration gradient. So when we talk about areas of high concentration and areas of low concentration, we're talking about a concentration gradient. This is a region along which the density or the amount of that particular substance either increases or decreases. These next few sections focus on passive transport. Passive transport is the diffusion of a substance across a biological membrane that has no energy expenditure from the cell. And I'm going to give you guys a little way to think about this in just a few moments. But we have two other types of passive transport we need to talk about first. The first is osmosis. Osmosis is the diffusion of water molecules, only water molecules, across a selectively permeable biological membrane. Water is one of those things that we need to live. Living things cannot survive without some type of water. With it being that important, water has its own type of passive transport, and we call it osmosis. The last is facilitated diffusion. This is the movement or passage of molecules through a selectively permeable membrane through specific transport proteins that still is not using energy from the cell. Now, I like to think of passive transport in this way. Passive transport goes from areas of high concentration to areas of low concentration with no energy expenditure from the cell. I like to think of passive transport as you're sitting on top of a slide, the very top of it. To get to the bottom of the slide, so the high area to the low area, you're not using any energy. Someone is going to push you down the slide and you're going to go from a high concentration to a low concentration. And it can occur in two different ways. The first is diffusion, which is just the movement of the molecules. So we can see here, the green section on this side has a lot of molecules on it that are going to pass through the membrane to the side that's more pink that doesn't have a lot of molecules concentrating it. So we're moving down our slide from an area of high concentration to an area of low concentration. It can also happen through facilitated diffusion. This follows the same procedure. We're just starting on the pink here. With our blue molecules, there's a lot on the pink, but we're going through a transport protein, no energy, still falling down our slide to our green area where there's very few. Passive transport will happen until the same number of molecules is on both sides of the membrane. Osmosis is the movement of water through a semi-permeable membrane. Water is so very important, and it can happen in three different ways depending on the cell. The first type of osmosis is called isotonic. This is where the concentration of water inside of the cell is the same as the concentration of the water outside of the cell. Meaning we have say 12 water molecules inside our cell and 12 water molecules outside of our cell. As one of our water molecules leaves the cell, another one will enter. Osmosis is balanced. Our second type of osmosis is called a hypertonic. This is a solution that has more particles outside of the cell than inside of the cell, causing the water to leave to try to balance out that concentration. And I'll show you this in one moment. A hypotonic solution is a solution that has fewer dissolved particles outside of the cell than inside of the cell, meaning the water is going to try to enter the cell. So here's our isotonic solution. We like to look at this in humans as red blood cells. We want our red blood cells to sit in isotonic solutions, meaning the same amount of water is inside and outside of the cell. As one water molecule leaves, another one can enter. A hypertonic solution means that water is going to leave the cell because there is more, say, sugar or salt outside of the cell, and the water wants to try to balance out and create that equilibrium again, meaning our cell is going to shrivel. We don't want our red blood cells to shrivel. If they shrivel, they can cause clots, which causes a lot of issues with the health of the organism. And a hypotonic solution is the opposite. So there are fewer dissolved particles outside of the cell than inside of the cell, meaning water is going to try to create equilibrium by entering the cell, meaning our blood vessels, our blood cells are going to swell like balloons, which again, we don't want when our blood cells blow up like a balloon or fill up like too much with water. They don't carry as much oxygen throughout the rest of the body, and they can do something like this one here, which is bursting, letting all that water out. Facilitated diffusion, we showed this on that passive transport slide. This is the movement across a semi-permeable membrane from areas of high concentration to areas of low concentration through the use of a protein channel. So when we talked about the cell membrane, we talked that proteins can serve as doorways or channels. When a molecule needs to pass through, it can happen in two ways. First is that it's like a tunnel that's always open and those molecules can pass in and out of the cell as much as they please. The second is that it must change shape. And this is common because it's going to make sure that the cell does in fact need that particular molecule. Facilitated diffusion, because it is a form of diffusion, is still going down our slide from areas of high concentration to areas of low concentration. The difference is they just need a doorway to do it. This allows molecules that normally would not be able to interact with those fatty acid tails of our phospholipid to easily enter and exit the cell without overwhelming the cell membrane. We are going to stop here and pick up with active transport in our next video.